Good morning or good evening, everyone, depending on what is your time zone at the moment. My name is Maria Schatz, and I'm a scientific program manager for the NIEHS Office of Data Science. It is my pleasure to welcome you today to the AOP standardization workshop sponsored by the Environmental Health Language Collaborative, ELC. And what started about 6 months ago is an idea to bring together a small group of people from AOP and knowledge graph fields, evolved into a much grander undertaking. We invited almost 150 people to participate, both from Europe and the US, spanning industry, government, academia, and regulatory agencies. And next, I would like to invite Charles Schmidt, the director of NIEHS Office of Data Science, to start us up. Is Charles.
Hi, everyone, and thank you for making it here today. In addition to directing the Office of Data Science, I work closely with Maria Schatz on co-chairing the Environmental Health Language Collaborative, which we call ELC. ELC is a community of practice that we started back in 2021, really with this mission of advancing, can you go back? Advancing environmental health research by promoting really common and harmonizable language so that we have standardized data sets and interoperable knowledge bases and tools. Okay, next. I should say ELC works primarily through a few different mechanisms. We hold webinars and different events to communicate out progress in this area and to discuss issues. We host working groups that try to advance solutions in different areas. And we host workshops like this one. And this workshop, as Maria mentioned, came out of conversations that really Holly had brought up in her role. She serves on the ELC Executive Committee. And we thought this would be a good opportunity to bring together the AOP community with members of other communities, really because we're at a junction point now where there is and has been significant money and investments going into a number of related communities. The AOP community, obviously, but the biomedical knowledge graph community has been getting substantial funding in recent years from NIH and NSF. The novel approach methodologies community has always been funded, but is receiving new money coming in through the NIH Common Fund program. We have grants, NIEHS in particular, and data sharing, data standards. Obviously, there's been an explosion in the AI and NLP biomedical communities, and there's been significant new funding coming into the exposome community. And I will apologize, I have a very US NIH-centric view of the world, and I apologize for that bias when I talk about things. But regardless, there has been a lot of new activities in these areas, and we want to make sure that we can foster the right level of communications and sharing across these communities, as well as share advances for curating, representing, and storing knowledge across these communities. Next. So we do have a charge for everyone that's here today. We do want to raise awareness of AOP community efforts as we progress as a community, both within that community, but with these other communities. We want to increase alignment and collaborations with related efforts, in particular with the biomedical knowledge base and ontology communities. It would be a shame to see these communities evolve using different standards that are not interoperable. And we do have some specific questions and are seeking advice to inform existing and future efforts that you'll hear about today. Okay, next. And I do have kind of a selfish reason, speaking from the NIEHS side. Obviously, AOPs are a critical component of environmental health and its knowledge ecosystem. We generate data and we generate knowledge. And I would like to be in a position where we can get that knowledge into AOPs as much as possible and automate it in semi-automated ways. We've been looking at this within the SED program, but we're really hoping that these communities represented today can advance methods around data formats, data models, the protocols, the terminologies, and how to represent quality of data, so that we can start automating these data collection systems that we have and that we know will be coming online in the future. So with that, I guess I'll turn it over to Holly.
Thanks, Charles. That's a great opening for our meeting here today. Hello, welcome everyone. I'm Holly Mortensen, coming to you from EPA Office of Research and Development. I'm also, as Charles mentioned, part of the executive committee for ELK, and with Charles and Maria have organized these couple of days with our co-chair and colleague, Jenny Hench, with open data biomodeling. So today I have a few slides, and I will come to you as an AOP tool creator mostly for these few slides, and talk to you about the FAIR AOP roadmap and some other community efforts related to AOP standards. Next slide, please. So just a quick overview of the presentation. I'll look at our timeline for AOP tool development and some of the other efforts sprinkled in there that you'll hear about throughout the next couple of days as well. I'll talk to you about the FAIR AOP efforts, focusing on the second FAIR AOP paper and the organization that came to be called the FAIR AOP Cluster Work Group. And then I'll talk to you about a couple of OECD proposals, or actually a few, and how they fit together, and round down with a call for papers. So not subliminal at all at the bottom of this slide are some of the areas of focus for us to think about today. So next slide. So our AOP tool timeline, I just wanted to bring this up for folks that aren't necessarily familiar with the evolution of AOPs so much in their work, and then for those of us that are, just as a reminder. So as some of us are well aware, the AOP Wiki came online in and about 2013-14, followed by the AOP database, which is what I helped to create. And it started as an internal only instance. Just the few things that I wanted to point out here for the AOP Wiki and the database. Some of the ontology annotation was happening around 2017, and the AOP database work focused specifically on mapping key events and key event components to protein ontology identifiers, and then to their respective gene IDs. So a lot of what I'll focus on in the next couple of slides have to do with that. And the 2021 paper that we had, which came out, which introduced the AOP database research description framework, or RDF, work that began as a collaborative effort between the US EPA and the University of Maastricht. So on this timeline, you see things sort of take off with the introduction of the AOP Wiki XML, which made AOP data available, and then the RDF mapping of the wiki and the AOP database, which made the AOP data parsable and interoperable. And so by 2022, we start to see real reuse of AOP process data and graph database and AI approaches start to come online, and the data start to be reused. For example, with CompTox AI and the AOP Network Finder is our most recent rendition, which pulls from a reuse of data. So next slide. So there's a whole lot of efforts going on, AOP-focused efforts addressing various aspects of AOPs. Though the OECD does not specifically endorse any of the third-party tools outside of, or tools in general outside of the wiki, there are several technical implementations, or referred to as third-party tools, which I'm pointing out here. And two important points regarding the AOP technical tools are that the AOP technical tools translate AOP data and metadata to improve the mechanistic content of AOPs, and how the AOP tools consume and manipulate the data from the wiki are dependent to a certain extent on how AOP and related data are structured within the wiki. Next slide, please. So just taking you back in time here, our current efforts with the FAIR AOP cluster, Penny will tell you more about how it came to be with the first FAIR effort. But what came out of that was a second effort forming the FAIR AOP cluster, as I mentioned, where we engaged tool developers and FAIR data advocates to address how to improve AOP data through coordination of effort and alignment of tools. So the second half of the slide is an old one, about 10 years now, from the AOP database, but I bring this up now because basically we're still asking the same questions as tool developers. Can we link AOPs to multiple data sources to understand the biological context of AOPs, so chemical, gene, disease, assay pathway, etc.? And can we improve the machine readability of AOPs? And then can we develop case studies to inform mechanisms? So these kind of questions persist. Next slide. So in this, our second FAIR paper that we referenced a couple slides ago, basically we consider the AOP to be an AOP tool along with all of the other tools, but the wiki is not really shown here in terms of a schema. But you do see the schemas for all of the various third-party tools. So with the point being that each AOP tool implements unique processes to store and map and update the data, or it reuses previous iterations, as indicated here by these little blue arrows. So our AOP Wiki RDF is feeding into the new AOP Network Finder. AOP database is the basis for what's in CompTox AI. And currently none of these tools are interoperable. All overlap in terms of their data domain and content for gene, chemical, pathway, etc. So next slide. This is a figure taken directly from the new FAIR paper that is in Zenodo now, and will probably be in CompTox shortly. But the data domain and coverage overlap is the main theme of this figure. And what became apparent really early on in the deliberations of the FAIR AOP cluster is that each tool developer had worked to map AOP wiki data and other biomedical sources of information. But I don't know, looks like it got a little bit jumbled here. Sorry about that. But a collective goal then is to really streamline this process and reduce repetition and make this task less onerous for researchers going forward, so that as a community, we don't have to keep repeating the same process in various tools and versions. Here you can see the repetition of effort with the domain overlap among the tools. And I'd like to underline at a time when resources are limited and coordination of efforts is really what's required. So the next slide. This is an adaptation from Schultz FAIR Hourglass, and we included this in the second paper to really show that our FAIR AOP tools are basically at this level of FAIR ready, which is they're machine actionable, but they're not interoperable. So we're kind of stuck in this FAIR ready state, making the Hourglass top heavy. But with this, our questions sort of shift, and we're asking now, can we harmonize and coordinate processed AOP data to foster interoperability and reduce redundancy, but also would this process allow increased insight into mechanistic content of AOPs? And what role does centralization of AOP information have in trustability? Next slide. So some of our future efforts and some of the presentations that you'll hear during this workshop are focused on the implementation of AI for risk assessment or regulatory activities. Issues of data provenance have been raised for the AOP Wiki, but I think it's also apparent that subsequent processing and reprocessing of AOP data has its own provenance and tracking issues, which persist. And so I just wanted to point that out. So we're still seeing bolstering across projects that are related or addressing regulatory implications, that AOP information is bolstered with disease to interpret mechanism. And it's my hope that if we're able to streamline and coordinate our processes for primary AOP data, that we can more confidently apply tools to interrogate variation mechanism, for example, and make use of AOPs more effectively for regulatory applications. The next slide. Just briefly, resolving the process and output from various third-party tools and directing back to the wiki is one area of interest. So our AOP Wiki evidence modeling framework prototype, which will be discussed by our next speaker, Dr. Jenny Hinch, includes some areas that could potentially be used to incorporate biomedical entities. And so the information available is directly coming from the primary repository of AOP information. Next slide. We have submitted an OECD AOP reporting standards proposal for mapping of biomedical information back to the wiki. The wiki is preferred centralized implementation of these data, but it does have an associated cost. So we'll be developing case studies and welcome your comments and ideas. So please do reach out if you have specific opinions about methods or repository locations, etc. There's two other proposals I'll just briefly mention, but you will hear more about one of those, at least here. Two other relevant ones. One from Julia, the omics to AOP, that focuses on linking specifically omics assays to AOPs. And the second one is the OECD Harmonized Templates effort that links AOP key events to chemical mechanistic effects and will is thought to be feeding into iEuclid in the future intended for regulatory community use. And so my last slide, I believe, or second to last, we are hoping to put the workshop report for these meetings into our special issue. I wanted to put out a call for papers, and direct all of your recent papers into this F1000 issue we've put together on AOP mechanistic and data-oriented collection. So we'd love to see your manuscripts. I think it accepts posters and abstracts as well. So pass this on to your colleagues and consider it the special issue for your own research. And lastly, I'll just reiterate the message throughout the sessions probably, but underline that everyone involved in these workshops to contribute your ideas, and they're all extremely important to improving our AOPs in the future. So with regards to potential outcomes, whether it be for the OECD case studies, improvements to interoperability, or manuscript contributions, I want to encourage you to reach out and get in touch. And then the last slide, I think it's probably a little bit early for acknowledgements, but there were a lot of folks involved in putting these next 2 days of workshop together. So I want to thank especially Charles and Maria and the EC for recognizing the importance of AOP standards work, and for my esteemed co-chair, Jenny Hinch. And I will let her take it away next.
Thank you, Holly. All right, so I think we might be a little bit behind, so I'm going to try to go quick. So for those who don't know me, my name is Jenny Hinch, and I am a developer for the AOP Wiki. I've also been involved in AOP development and applying AOP framework principles to different efforts. Right now, I'm going to give a high-level overview of AOPs for some of the people who might be new, since we've invited people from ELK and the biomedical research community. And then I'll introduce some of the key concepts associated with the AOP Wiki evidence modeling, or EMOD work that Holly just mentioned. Next. This is my disclaimer slide, which I will skip now to catch up on time. Thank you. So the AOP is a conceptual framework for organizing biomechanistic toxicology data, information, and evidence. Next. And the first step involved in AOP development involves scoping out the purpose of the AOP with respect to risk assessment decision-making. Next. And next, I'm not gonna read through all of these. One more, please. Yes. Okay. So I do want to highlight that AOPs provide context, biological context for alternatives to animal testing or new approach methodologies referred to as NIEHS. And as Holly mentioned, the AOP Wiki and its underlying database, the AOP KB, serve as a central repository for AOPs. Next. The developers of the AOP concept drew from a systems biology perspective and have since proposed guidance around AOP network development. Next. An important guiding principle in AOP development is that key events and key event relationships should be designed and developed for reuse across multiple AOPs, and that is to help facilitate AOP network development and reduce the time associated with developing new AOPs. Next. All right, so just to give a little bit of an example of the breadth of domain coverage, I've provided these two AOPs. One is a skin sensitization AOP, and the other is an AOP for aromatase inhibition in fish, leading to reproductive dysfunction. Both of these are associated with test guidelines that are used for risk assessment purposes. Next. So on a conceptual level, we can think of AOPs as a bridge between biomechanistic toxicology and regulatory science. You're going to hear more about some of the regulatory aspects this morning. But one of the biggest challenges in the AOP field is the amount of time and labor that's associated with generating evidence-backed AOPs. And so one of the questions that motivates me is thinking about how can we reduce this burden by enhancing the computability of AOPs in a way that accelerates development of robust evidence-backed AOPs. Next. So there's a few different ways that we can do this, different resources to leverage, and a lot of these will be covered today and tomorrow in this workshop. What I'm going to talk about is how we can think about improvements to the AOP Wiki infrastructure as a way to enhance the computability of AOPs. Next. So when we think about evidence in the context of the AOP framework and the AOP Wiki data model, we're interested in the key event relationships, which I've highlighted in this graphic. We want to know the available weight of evidence to causally link an upstream event to a downstream event. Within the wiki, we've got 7 different evidence fields for KERs, all of which are unstructured free text fields, which are burdensome to aggregate by those who are AOP developers and also burdensome to read through by people who are doing the review. And so determining a way to structure information within the AOP Wiki would significantly advance the computability of AOPs and make AOPs more fair, findable, accessible, interoperable, and reusable. Next. So this is where the EMOD or evidence modeling prototype work comes in. Next. So the guiding principle established in the first EMOD prototype, which has been carried forward with each iteration we've built upon since, is that we're structuring empirical weight of evidence information using computer readable ontologies and control vocabularies as much as possible. With EMOD, we introduced new data objects, which are shown here in gold. The observation and evidence objects are the two main ones. So an evidence object relates to a KER, and it has 2 child data objects, which we call the observation data objects that relate to the key events. So here on the right, you see the observation fields, and this is where we take advantage of some of the properties within the AOP Wiki data model that are already structured. So you see that we have taxa, species, and life stage listed in this. This is a screenshot of the observation form, and we were populating that form with existing tables that we have, that some of which do map to other controlled vocabularies and gene names and species, or sorry, not gene names, but species lists. And then we also incorporated elements of the event component, which I will talk about more this afternoon. Next. So the data model that we just looked at for eMod version 1.1 enabled us to build this evidence map UI feature, which offers a visual summary of the weight of evidence. We wanted to offer AOP consumers a way to quickly assess weight of evidence scores in a high-level summary manner, while still having the more detailed empirical evidence information available, which is stored in the data object models that I showed. So before going to the next slide, I want to acknowledge that Jason O'Brien and Steve Edwards were the ones who first developed the specifications for eMod version 1.0 and secured funds for the work through ARDF, the Alternatives Research and Development Foundation, and CCAAM, which is the Canadian Center for Alternatives to Animal Methods. Next. So in the latest iteration of the eMod model that you see here, and this aligns with what Holly showed in her presentation, it does provide a structure for biological elements that we can map to ontologies and controlled vocabularies. The purple box shown here for assays was introduced as part of the Methods to AOP collaborative group, which Helena Hogwark-Durdock will talk about later this morning. Later this afternoon, I will provide a little bit more details on the eMod work, and we'll be inviting your feedback during the breakout session. So next. In closing, I'd like to acknowledge the funding organizations that supported the work I showed here. The European Commission Joint Research Center has funded AOP wiki maintenance and development since around 2019. ARDF and CCAAM funded the first eMod prototype, and the Endocrine Disruptor Screening Program at US EPA funded some of the most recent eMod iterations. Thanks for listening.
And next we have Penny Nymark from Karolinska Institute. Penny, can you please share your screen?
Yes, I am trying to do that now. Can you see?
Yes, we see it in a kind of slide. Yeah.
Like this?
Yep.
Yeah, great.
Looks good, Penny.
Okay, thank you. So my name is Penny Nymark. I'm an assistant professor at the Institute of Environmental Medicine at Karolinska Institutet. I can also mention that I'm a regulator actually at the Swedish Chemicals Agency. So I have some regulation background as well, but I will be talking to you as a researcher at KI. So I'm going to talk about what we refer to as the first FAIR AOP paper. And I'll go a little bit into some experience. I won't have time to talk that much about the experience of implementation in the EU, but I added that title just because I'm going to touch on the subject a little bit. And so I'm going to start off with this figure that I like really a lot. It's a figure that was shown by Nicole Kleinstroyer a couple years ago. And I like it because it really points to the FAIR resource as being very central in the predictive toxicology vision that we're all hoping for, sort of. And so, and in addition, AOPs are, of course, a part of this circular improvement of all the models and data that goes around and improves risk assessment, or so we hope. And so these two really build the core, or are the cornerstones, I would say. AOPs actually, I would say, deserve a bigger place in this figure, but fairness is certainly a big part of it. And a big aspect of this is, I don't know if, can you see the whole slide now? Because I have something in the way. Can someone confirm?
Yes. Yes.
Okay, good.
You look good, Penny.
Yes. Good. Thanks. So fair big data and AOPs really enable explainability and overall trust. And I'm going to try to make that point on a conceptual level. I'm not going to be that technical as Jenny and Holly were, but I'm really happy that they gave a very good basis regarding both the history of fairness of AOPs and the AOPs themselves and how the AOP wiki works. But so trust is the central aspect of my talk. And but what are the FAIR principles then? And I want to start with saying that the FAIR principles are not really something new. They're actually something that was designed by the informatics community to basically harmonize efforts that had already been in play for a long time, and basically since the start of internet and even before that. So the FAIR principles really are not or the principles are new, but the idea behind them and the solutions that fairify data are not new really. But so these principles basically consist of the 4 words that many of you have probably heard already: findable, accessible, interoperable, reusable. But then underneath those are a number of sub-principles, and a big part of those focus on metadata, and in particular, machine actionable metadata, which Ginny was referring to, and Holly as well, a lot. And this, of course, matters because without the data that explains the data, we don't know what the data is. So metadata is extremely important and also plays a large role in the interoperability between different types of data. And so, but in addition to that, there's also 2 aspects that are important to the FAIR principles, and the first one being the technical infrastructure that comes with some of the FAIR principles. So, and with that, it refers basically to the generic solutions and services that basically allow for machine actionability. And so, for example, globally unique identifiers or standardized communication protocols, which are all things that the internet basically implement. The other part is the more difficult part, and that kind of relates strongly to the AOP wiki and the AOPs, is the social contracts and the domain-specific agreements, basically, that need to be taken in order for the FAIR solutions to work for that domain and for the fairness to become meaningful, so to speak. So when it comes to AOPs, what we did at the start of the FAIR AOP cluster was to take this picture, which Dries Knappen published a few years ago. And I like the way how it describes basically how we have moved from the AOPs originally being these pragmatically simple entities, but which had very low utility, largely because of the lack of trust and lack of detailed data and transparency in these frameworks. And how we have now moved to a level of necessary complexity, which details the AOPs better and explains them better, but they're still low utility because it requires expertise. Now, what we envision then the FAIR AOPs or fairification of AOPs to do is to really take us to the informed simplicity where high utility suddenly becomes, or they basically become high utility entities because of the underlying data that enables machine actionability. And so this was the basis for our first paper, so to speak, on why AOPs need to be fair. And basically what the sort of outcome of that paper, we had much more higher ambitions at the time, but in the end it was a lot of discussion and a lot of challenges to really understand what we mean with fair AOPs. And we had a strong focus on the fact that fair AOPs allow for intuitive visualization, inclusivity of data and methods, increased trust, and basically today, as we know, AI is increasing explainable AI-driven decisions. And I'll try to point to this also throughout the next slides. So what do we then mean really with social versus technical fair implementation in the AOP wiki? Well, on the social side, and you might have seen this hourglass that Holly showed earlier, basically the point there is that the social verification can operate by itself within the domain and take decisions, for example, on which controlled vocabularies should be used in order to, sorry, I need to drink some, best represent the community of AOP developers and users, and to support interoperability with AOP relevant external data resources. And on the other side, you have the technical fair orchestration, which is then the question of whether or how the AOP wiki data model should handle the selected controlled vocabularies in order for the AOPs to be broadly represented in the context of all data. So in the internet, basically, and I think that's one of the most central aspects of fair AOPs. They should be interoperable with everything else. That sounds maybe like a big vision, but that's what you expect from a machine actionable data. And also they should be interoperable with other AOP wiki third-party tools, which utilize diverse standard communication protocols, for example. So that's that was kind of, a little bit of an example of what fair really means in terms of the AOP wiki. So, but then why I'm talking so much about trust, and I guess, I mean, it's a given that you want to trust something that you're going to use in regulatory context, but what really matters is that regulatory risk assessment depends really on trust, and the way that comes through is that it's dependent on data that is validated, or methods that are validated, and especially highly, at least in the European Union. And I say this as well, I'm a little bit biased in terms of how I look at the regulatory frameworks because of being in the EU. But a big part of that is the mutual acceptance of data, which basically allows for both reduced amounts of animal testing and also allows for resource and process efficiency, and overall strives for the legal certainty that is necessary in regulatory risk assessment. But then on the other hand, you have also something in the EU that is has been recommended by the Commission at the moment, which is called Pre-Market Safe and Sustainable by Design, which is a risk assessment that happens before regulatory or law kicks in, basically. And that side equally depends on trust, but it's a little bit more flexible in terms of legal certainty and is more dependent on scientific confidence. Of course, it also requires or strives for having data that is applicable for the later regulatory requirements. But these two aspects are places where AOPs are starting to pop up and make a difference, so to speak. So I'm going to just briefly, before I not sure if I'm going over time, but I have just 2 slides, I think, left. Just briefly say that in the EU, there's an active shifting towards the paradigm of phasing out animals. And one part is the EU roadmap of phasing out animal testing for chemical safety assessments. And those discussions are being held on very high policy level. But what you notice from, and I also want to mention the other aspect that is, or the other initiative that is ongoing, which is the European Test Method and Validation Strategy, and which is not directly striving for phasing out animal testing, but is looking at the development of any kind of test method useful for regulatory assessment. And both these efforts are strongly coupled to data, naturally, and you can see that already on the left side, that the EU is setting up a data platform, which is kind of in line with the predictive toxicology vision, where data is the essence of the circularity and how everything works. And that is aimed, of course, then for faster development of effective models. And now you don't really hear a lot yet about the AOPs in these efforts, but they do pop up on the side just because they are so pragmatic for the purpose of describing and putting together data. It's still the data that matters, but the AOPs are being used strongly as a support. And of course, the fair verification of AOPs can be expected really to accelerate and allow for better and more trusted models development, and coupled as well to the validation strategy that naturally has a big part as well in terms of leading test method, or allowing test methods to become a basis for mutually accepted data. So we slowly see the AOPs sipping into these efforts, but they still require better interoperability with diverse tools and methods that show up in these frameworks. Now, on the other side, on the other hand, basically, the safe and sustainable by design framework, which is a bit more flexible when it comes to implementation of NIEHS, new approach methodologies, and other types of new data that are not normally accepted within regulation. This is a paper that we've been working on, or we were working on within the PARC initiative, and we're continuing now within the PINK initiative. And the AOPs really play a big role in terms of allowing for so-called seamless, fair data and tools-enabled assessments during the innovation approach. So that's what safe and sustainable by design really is about, allowing for risk assessment to happen early during the innovation process and allowing for collection of data. And naturally, the AOPs serve as very good transparent platforms for increasing the trust that is needed also on the SSBD side in NIEHS data. And so I had a final slide, but I'm going to skip that, as it was basically pretty much, I wanted to say that when fair, the data is always there. I found that funny because I wrote it down and it rhymed. And I really think that that basically is the essence of the trust, that when you have an AOP that is fair, it's also machine actionable, which means that you can dig into the data if you want to. You don't necessarily have to, but the trust is built on the fact that you know it's possible and someone else might be able to dig in and provide evidence of quality and trust in that way. So with that, I want to thank you, my funders, and a whole lot of collaborators. I didn't put them all on the slide, but there's a lot of people behind all of the work that we've been doing within these projects. And I also want to point to a new third-party AI-driven AOP tool that I just recently had fun with together with some collaborators, and which is not among the third-party tools in the AOP wiki. We also had a webinar a few years back, or a year, or maybe 2 years back, which is available on YouTube if you want to know more. And the upcoming publication by Holly, which is a really nice paper. So thank you.
Thank you very much, Penny. And Scott, you are next. And we will spend a few minutes for Q&A after Scott's talk.
Okay, can you
You're good to go.
see my slide?
Mm-hmm.
Great, and you can hear me. So thank you for inviting me to come and talk. My name is Scott Lynn. I work in the US EPA for the Endocrine Disruptor Screening Program. And I'm going to talk to you about how we're applying AOPs within our program. The presentation and views expressed by me do not necessarily represent or reflect US EPA policy. The Endocrine Disruptor Screening Program was established with the U.S. Food Quality Protection Act in 1996. The requirement within that legislative law, or within the law, was to for the EPA to screen pesticide chemicals for their potential for estrogenic effects in humans. There were a number of discretionary authorities, such as expanding for thyroid, or for androgen and for thyroid, and also to expand to wildlife, and also for screening of other substances outside of pesticides, particularly those that are found in safe drinking water. So I want to highlight something, and that is that unlike a lot of other risk assessments, within the Endocrine Disruptor Screening Program, we have what I would call the luxury of knowing exactly what mode of action we're interested in, and therefore having what I would consider to be a finite set of molecular initiating events. So in a way, this makes our program perfectly situated to utilize AOPs. So the EDSP has a tiered approach to screening chemicals. There is a tier 1 battery of assays, and these are 5 in vitro and 6 in vivo screens. The in vivo ones are indicated with the maroon text. The purpose of tier 1 is to detect the potential to interact with the endocrine system. And then tier 2 is called in only after a weight of evidence is performed on the tier 1 data. The tier 2 tests are multi-life stage. They're usually, or most of them are multi-generational studies. They cover a broad range of taxa, mammal, fish, amphibian, and avian. And these ultimately determine the adverse apical outcomes from the endocrine effects. So this slide illustrates which assays across the EDSP tiers have endpoints associated with the relevant endocrine pathways. In the left-hand column here, we have 5 endocrine pathways: estrogen agonism, estrogen antagonism, androgen agonism and antagonism, and thyroid. And across the top of the table are listed all the tier 1 and the tier 2 assays to show the endocrine pathways upon which each assay provides information. And these are indicated with these black squares. When we look at assays that will inform on estrogenic effects in humans, which is the legislative mandate that we're pursuing right now, we can identify 6 tier 1 and 2 tier 2 assays. The assays in purple inform on estrogen pathway, but these are performed with ecologically relevant species and are therefore not considered to be applicable to human health risk assessments. So in this slide, I've listed the generalized endpoints on the left here from all the assays, and then I've crosswalked them with the specific assays across the top. The in vivo endpoints and assays are shown with the dark red text again. And when we look at this list of endpoints, we can see a biological hierarchical pattern in which can be aligned with the general AOP nodes. And you can see them here going from MIE through cellular response, organism, and up to population level adverse outcomes. We can take this a step further and identify specific AOPs with estrogen receptor alpha activation as the molecular initiating events and key events or adverse outcomes that track to endpoints associated with the EDSP assays, such as AOP 167, which goes from estrogen receptor activation to uterine weight, and AOP 445, which goes from estrogen receptor activation to changes in fecundity or reproduction. Here I've mapped out these two AOPs and showing the distinct nodes and the general level of biological hierarchy at which each key event or adverse outcome resides. So if we have data that provides information that shows the chemical has estrogen receptor alpha activity, by applying these AOPs, we can potentially tie that bioactivity to the key events or the adverse outcomes of regulatory interest. So I'm going to shift gears a little and briefly describe the ToxCast estrogen receptor pathway model. The estrogen receptor pathway model incorporates data from 18 in vitro assays that cover a number of different nodes in the estrogen receptor pathway. These can be binding, dimerization, DNA binding, protein production, and ultimately ER-induced cell proliferation. The pathway model uses a model to integrate all of this data, and then it produces an area under the curve score, which gives a general sense of the potency of any chemical to activate the estrogen receptor pathway. The model outputs were evaluated against reference chemicals, and we took this to our science advisory panel in 2014. In 2023, the EPA deemed the estrogen receptor pathway model validated for use as an alternative for 3 Tier 1 EDSP assays. And these are shown here: the estrogen receptor binding, the estrogen receptor transcriptional activation, and the uterotrophic assay. Just as a point, the law actually, the FQPA requires the endocrine disruptor screening program to use validated assays in order to perform our screening. So establishing what is validated has been something that we've been working on for a while. This slide now shows the Tier 1 assays, but it indicates the assays for which alternatives have been validated and also provides the proposed models for alternatives for the remaining EDSP assays and tests. So we see up here at the top, we have the binding, the transcription, and the uterotrophic, for which we're using the ER receptor pathway model. We've also accepted the AR pathway model for AR binding, and we have, again, proposed model alternatives here for the rest of our assays. What we're showing here is the chemical space coverage for the full ER and AR pathway models. There were over 1,800 chemicals tested in these models. However, the important point on this slide is that we have data on estrogen receptor and androgen receptor activity for over 300 pesticide actives and 200 pesticide inerts. So with bioactivity data that supports an estrogen receptor activation molecular initiating event, we can use those AOPs to identify key events or adverse outcomes that track to the endpoints associated with the EDSP assays. Based on those AOPs, additional in vivo testing with the endpoints associated with those key events or adverse outcomes can then be requested, and that's exactly what we did. In October of 2023, about a year and a half ago, the EPA announced a strategic plan to advance the efforts of the EDSP to ensure that pesticide decisions continue to protect human health. One of the actions the EPA adopted was the strategic plan was to use ToxCast, estrogen receptor, and androgen receptor pathway model scores to identify which pesticides may potentially cause adverse endocrine activity through the human estrogen and androgen pathways. Looking at this flow chart on the right, EPA has laid out a framework for prioritizing pesticides that require additional in vivo data on potential human estrogen and/or androgen effects. Following the flowchart, you can see that the EPA identified 30 high-priority conventional pesticide active ingredients for additional Tier 1 testing. Those test orders began to be released, I think, in March of 2024. And I believe as of last month, they've all been issued, those data collection requests. The numbers have changed slightly. There was a reevaluation of some of the pathway model scores, and that caused some changes. So, wrapping up, because I realize I'm over time, revisiting the endpoints obtained with the EDSP assays and tests. While we have AOPs for the uterine weight and the generic AOP for reproduction, there are limited AOPs with key events or adverse outcomes that match the endpoints associated with our female pubertal assay, which I'm now showing here, circled. We've already begun developing AOPs linking the estrogen receptor activation to those endpoints associated with that assay. And once developed, those AOPs will allow us to identify assays that measure the key events in those AOPs, which can be an important step to developing NAMs or group of NAMs that may provide alternatives for the in vivo assay. The idea would ultimately be that we can have NAMs and in silico or high throughput approaches to more quickly screen the pesticide actives that were required to screen by law. And so the hope is that we can use the recent advances in ontology and AI to help us in this process. So that's it. Thank you very much. I want to thank other people associated with our program.
Helena is next. Helena, can you please share your slides?
Yes. Looks good. Okay, so I'm going to try to stay within my 12.5 minutes. I'm Helena Hogberg. I am the acting director of the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICEDAM. And our main role is to support ICVAM, which is the Interagency Coordinating Committee for the Validation of Alternative Methods, that consists of 15 US federal agencies that are all using toxicity data for regulatory or research purposes. And our focus is on developing, evaluating, and promoting new approach methods or NAMs for regulatory use. So why is the AOP framework important to NICEDAM and ICVAM's work? Because it can help to build confidence in using NAMs data to characterize risk or hazard. And recently we published the validation, qualification, and regulatory acceptance of NAMs report. So this figure is in there, which is describing key concepts to consider during development and implementation of flexible fit-for-purpose NAMs validation strategies. So we're trying to get the approach more flexible, because depending on the context of use. And I'm not going to go through all these key concepts, but I'm going to highlight the biological relevance, because this is where the AOP framework fits very nicely in. So biological relevance is basically to find similarities between the physiology of, or the biology measured by the test system, so the cell system or the NAM, and human biology. And you can do this by consider human dosimetry modeling, or what cell types that are used, so what cell types are in your model, and also the structure of the target organ tissue. But another way is also to anchor them to a NAM, to an established AOP through a MIE or a key event. And one of the most successful stories here is the OECD issued guideline 497, the defined approaches on skin sensitization, which was the first international harmonized guideline to describe a non-animal approach that can be used to replace an animal test. And NICEDAM was very heavily involved in this, and we evaluated performance that suggested actually that the defined approaches are overall better predictors of human sensitization hazard and potency than the animal in vivo test. And you can read about that in this report. So other things and other topics, or other fields where NICEDAM are using or incorporating the AOP framework, is In this example here, where we were collaborating with pharma to enhance seizure liability assessment of compounds. So what we did here was that we acknowledged that harmonizations or ontologies are very important for the AOP framework. And we used here the third-party BioVista visit tool in the AOP wiki to develop a network of AOPs that can lead to seizure. And you can see those here. Those AOPs are not so well connected, actually, because the key events were not standardized, so they were all using different terms or different ontologies when they were describing this key event. So what we did was we did a manual creation of these and build a more simple network that could be used to identify NAMs that then can measure these key events. And we are doing that by looking into databases. We have our own database, EIS, the Integrated Chemical Environment, that includes a diverse in vitro assay inventory, so mainly from TOX21 collaboration and EPA's TOXCAS data. And we have a lot of data across large chemical libraries here. And we are annotating these assays to mechanistic target using ontologies from OBO. And so we can, in this case, for the seizure project, for example, we could go in and look at these annotations and could then search for in vitro assays that could inform on the key events in the AOP network. And we have done similar approaches for other endpoints, such as developmental neurotoxicity, for example. So that's going to lead me into why those methods then are very important, and therefore we have the methods to AOP collaboration, which is what I'm going to spend the rest of 5-6 minutes to talk about. So this was an international collaboration, or it is an international collaboration with a lot of different people from different stakeholders, and many of you are here today. So if you don't see your name here and you were involved, please let me know, and I'm more than happy to add you to this slide. But as you can see, a lot of people. So why did we establish this? Why did we establish the methods AOP? Well, we all know, or we should know, that the key event in an AOP must be measurable, and therefore the methods and the information about it has a crucial role. But currently in the AOP wiki, this information is just a free text. So it's this box here, it's how is it measured or detected. So the author can fill in whatever they want. And even if you're looking at the AOP's developer's handbook, it actually says that this section is not intended to provide detailed protocols. But in this collaboration, we discussed that we think it's important that the information is available, and that it's for the test methods, since they are so important for, especially for promoting NAMs. It also creates inconsistency, and it does not really allow to compile and sharing structured information. So what did the methods to AOP do? We started to develop a formal proposal for the integration of structured methods documentation, including both the test detection system, so cell system or animal, and assay endpoint readout in the AOP framework. And this is what this little figure here is showing, that for MIE, for example, we can measure that with the method A, which then consists of these parts. Key event 1, methods B or method C could be, method B and method C could be used. Method C can also be used for key event 2 as well as method D. So you understand a little bit of the structure. So the idea behind this then is that this would migrate towards a more computable and comparable knowledge representation of methods information. It will also enhance tracking and transparency, and which then in the end could bolster regulatory confidence because they would have the information available. It can also present an opportunity for third-party tools to create more elaborate offerings that would include assay methods details if it's machine readable and in the AOP wiki. So what we did was we identified different information of the methods that should be included, and then we organized and standardized that information so it's not should just be a list of information, which makes it a little bit complicated and more challenging to fill out. So the first thing you would do is to click on the study type. So if it's in vitro, for example, this blue box will come up, and we're trying to standardize it between the different study types. But of course, you will have different information depending on if you're using in vitro or in vivo. So these are then these boxes that have different fields that you can fill in. So documentation, for example, SOP or publication, species, host species, target species. The subject type would be the cell type or the culture type. Care would be culture conditions and passage number, for example. And we also have limitations, which could be, for example, applicability domain. And then the stressor, as you can see, is outside of this, but we think that it can be included. Of course, an AOP is not should be stressor agnostic, but in this case, you could, for example, include reference compounds that you've used for your methods, which would also be very useful for a regulator to feel confident that the assay is the method is measuring what it's supposed to measure. And then we have assays separate, and this makes it easier if you want to, for example, link these, or you want to reuse these blocks. So you would have them separated, but still somehow connected. So you could use the first thing you have to do is to input your sample, which comes in this case then from the in vitro system. It could be cell lysate, for example. And then you have again the documentations, which would be, protocol links. Measurement would be endpoint molecular targets, and then characteristics would be more providing the detection method and instruments and more details on those. And then we also discussed in the methods to AOP collaboration, how could this look like if we want to try to incorporate it into the AOP wiki? Our role was not to put it into, it was just more coming up with a suggestion. And I know Jeannie will talk a little bit about how she sees this could be incorporated eventually. So basically what you see here is the gray box is what the methods to AOP has what we are proposing to the methods information, and you can see how it's linked to each other, and we're trying to use ontologies. And then the things that are outside of the box are things that are already structured very well in the AOP wiki. But of course, the methods information would feed into those different blocks. But to do this, we realize we do need to broaden the AOP community, because several of you have developed AOPs, and we all know it's a heavy time-consuming process. And if we would require additional methods documentation, that would be even more of a burden to the AOP developer, authors and developers. And therefore, we suggest that we instead try to engage methods developers to contribute this information, because they are the ones that actually has easy on hand and know their methods much more than what you would find in the literature, for example. Another thing that this could help with is to promote test methods and scan for underserved key events. So for example, if a key event have been defined, but there's not so many suitable test methods, then you could see that gap, and that could be an opportunity for methods developers to develop new methods. And it could also be an opportunity for funding, for example. If we see that those methods doesn't exist, we could try to get funding to develop those kind of methods. So this is my last slide, which is more take-home message and future needs for if we want to implement this. First of all, the take-home message is that if we would incorporate robust, standardized, and controlled vocabularies for methods information, it could help to demonstrate and assess relevance of methods' ability to inform on a key event. It would build transparency and increase the confidence in AOP knowledge, because you would know how you can get this kind of information. It would also promote interoperability and reuse of both the key events and the methods, because you have more than one way to link them, and provide a resource for finding established methods when designing in experiments. Here we're thinking mainly about maybe basic scientists that have a question, and then they want to try to understand what method they can use to measure certain things. They could go into have AOP Wiki as a resource also for those. And then, of course, if we want to implement this has future needs. First, we need some training and communication to engage the methods developer and the AOP author, so they really feel confident and want to incorporate this information. We also realized that we probably want to try to harmonize the methods information documentation even more with some of the existing other documents, so OECD test guidelines, readiness criteria, and harmonized templates, for example, OHT 2.1 is one example. And then we proposed the initial stage of this kind of information, but we do want to develop case studies to evaluate and refine the suggested methods information, because we want to, of course, ensure that it's easy and user-friendly approach. And it might look different for different case studies, I can imagine as well, but that's one of the steps where we would like to go and try to really test the system. And then, of course, again, prospective changes to the AOP knowledge base needs to take place if we want to incorporate this information. So with that, I'll thank all of you, and I thank my NICEDAM group and the Methods to AOP collaboration. And I do see that I have a question in the chat.
Helena, we will take questions later after
Okay,
Madison
good.
finish. Thank you so
Sounds
much.
good.
Yeah, Madison, you're next.
Great. Well, hi, everybody. So my name is Madison Feshuk. I'm a biologist here at the US EPA's Center for Computational Toxicology and Exposure. And I'm excited to share this presentation today to tell you all a little bit about how the ToxCast program is attempting to connect to the AOP framework to support assay interpretation and data use. So for today's presentation, I'm going to start with a brief overview of the ToxCast program, which has already been alluded to in a few presentations, and then I'm going to dive into some of our assay annotation practices, as well as some ways that we're connecting to the AOP framework. And as a disclaimer, the views expressed in this presentation are my own and do not reflect the views of US EPA policies. So with that, what is ToxCast and why is it important? So as some background, number of limitations to our traditional toxicology methods for risk assessment, not enough time or money to generate all of the traditional animal-based hazard data out there for all the chemicals and combinations. So EPA really needs to think about rapid and efficient methods to be able to prioritize and evaluate for the thousands of chemicals that are out there in commerce. So given this challenge and imperative, to move away from our traditional animal models, we have the computational toxicology blueprint that attempts to outline this tiered testing strategy for hazard characterization using different new approach methodologies. So these NAMs can use computational models based on chemical structures or properties, or can employ different in vitro assays. And the gist of the blueprint is to first try to broadly prioritize which chemicals will then warrant more targeted and confirmatory screening, such as within a ToxCast assay, which are highlighted here. So we can put the CompTox chemicals blueprint in context of an AOP by considering the biology. So our broad profiling NAMs, like our transcriptomics and phenotypic profiling, will begin to help us understand how cellular processes may have implications upstream or downstream. But ToxCast assays, which are heterogeneous and more targeted, really will dial in and solidify our understanding of the biology by looking at a range of different specific event types, which can all map out to different places along the AOP framework. So what is ToxCast? So when I refer to the program, it's going to encompass our pipelining software, TCPL, along with our dependency package, TCPL-Fit2, our database of bioactivity data, known as InVitroDB, and then all of the curation efforts that go into processing the data and curating it to make it informative for our users. So I don't want to dive too deeply into our process that we have for adding new data into the ToxCast database. I'm going to encourage y'all to check out our vignettes, which have a lot more documentation out there. But ultimately, after all of the processing, QC, and curation that goes in to this, the ToxCast team aims to release data annually, at which point the public can go and explore data. And there are a few main avenues that most users are going to do. So we have, of course, data available via the CompTox chemicals dashboard in the bioactivity module. We also have computational toxicology and exposure bioactivity APIs. And then we make all of our data, so like the MySQL database and other flat summary files available on our downloadable data page. So with that background, we can dive into some of our annotation and AOP practices. So just to put the size of the database into context here, so our In Vitro DB 4.2 release, which came out in September 2024, includes aggregated data from 26 different assay sources, including the Tox21 Federal Consortium Program, 655 different assay platforms, and 1,570 normalized assay readouts, which we refer to as assay endpoints. This data covers a diverse biological space with over 500 mapped gene targets, and reflects different assay designs that will look at different cellular signaling pathways, protein interactions, and more complex processes. So to really make sense of this data, given the diversity of assays included, annotations are going to be necessary to describe all the different aspects of the assays. So our database was originally constructed based on the bioassay ontology framework and has different annotation fields to capture different types of information, where each annotation is going to be assigned as a feature to each of the different assay element levels. So we have our assay source, so generally who conducted the assay, what platform was used, assay component, so a raw readout of what was measured, and then assay component endpoint, so how that measurement can be interpreted, also known as the normalized component data. So some of our most useful annotations may be the tissues or cells that were used, as well as our intended target field. So a lot of our annotations are going to be employing different controlled vocabularies. On the left here, we have a pie chart of the tissue field. And then some of our annotations are also going to be hierarchical. So as an example, we have our general and intended target family, which also has a more specific intended target family subfield. And that's visualized, the intended target family field is visualized in that word cloud there. And I just wanted to highlight that field because for a number of assays, this is going to be a very useful annotation to consider, given that not all assays will have like gene mappings, for instance, but instead will map to larger processes. So in addition to all of the assay element annotations that we have a number of auxiliary annotation tables that capture additional information. So we have a number of assay lists that we curate. We have linkages to relevant gene identifiers from NCBI. We have a table to store different reagents and experimental conditions that were applied. We have linkages to different publications that are either describing the, oops, sorry, I don't know why it's doing that, describing the assay design or results. And then as a new addition for In Vitro DB 4.3, which is hopefully coming out sometime this summer, we're going to be including relevant linkages to the adverse outcome pathways and key events. So for 4.3, we've updated our intended target table so that now we have it as a long format storage to include mappings across different target types. So for past versions, our intended target table only stored gene mappings. Now we've updated that table to store gene, AOP, and key events, but we could definitely, grow it to include other things in the future, given the flexibility of the storage. And so here's an example of the table joined with some gene metadata. And just to note that currently ToxCast is only supporting mappings to the OECD approved AOPs, but in the future we might add some provisional mappings based on integration efforts with the AOP-DB project. So just to summarize the coverage a little bit, so for In Vitro DB 4.3, about 72% of the endpoints in the database have at least one gene mapping. And this represents 577 unique genes. When we look at the AOP and key event mappings, about 14% of endpoints have at least one. And this is representing 37 unique AOPs and 29 unique key events. And I would like to acknowledge Travis and Dan for working on this curation effort. I'm really excited to see this information database now moving forward. So next I want to highlight, you know, the data is in the database, but how is it also accessible in other tools? So here I just wanted to highlight how you can access this information via the CompTox Chemicals dashboard. So within our ToxCast bioactivity summary grid, we present a view of the potency estimates and annotations for ToxCast endpoints for a given chemical. And if you, we also have two columns there for AOP and event that have been curated by the assay endpoint that will link you out directly to the AOP wiki for more information. Similarly, if you move to the executive summary tab for a given chemical under AOP information, you can find links out to the AOP wiki, which essentially just provides a string of all of the AOPs that have been pulled based on the ToxCast mappings presented. So as a future idea, maybe for things to come, we think it'll be beneficial to integrate AOP searching into our dashboard. So similar to how assay gene is an option currently, the idea here would be that AOP and key event searching would essentially allow users to search for the string or term that's used in the title of these AOP or key events, and then return the list of relevant ToxCast assays. So finally, I just want to note that these annotations are great to help us flexibly aggregate and differentiate data, but there are definitely situations where we want our users to review the assay and seek out this more comprehensive assay documentation. So within the CompTox Chemicals dashboard, we do have currently some guidance document 211 aligned assay description documents available, and we have plans and these what's currently available is an older version, but they're going to be updated shortly. And then there are plans for the next iteration of these reports to include all of the new data-based AOP and key event mappings within the report, just so that it's one comprehensive document describing the method. And so that's just a visual of where it is on the dashboard. You have to add the column. It's currently hidden by default. But once you pop open the PDF, you'll be able to just see the document for the given endpoint that you're interested in learning more about. So yeah, just to, I guess, summarize my talk with a few bullets. So the ToxCast program makes targeted in vitro screening assay data publicly available for prioritization and hazard characterization. These efforts for more comprehensive assay documentation aligned with international standardization efforts like this one, as well as integration into the AOP framework, can definitely make this data more useful, interpretable for a variety of research and regulatory applications. So with that, happy to take questions, and thank you for listening.
All right, thank you so much, Madison. I see we have a couple of questions for you, and then one from for Helena. If you can read the chat, so please go ahead and answer the question as you're comfortable. Since that was a short session, I believe we can skip the break, and after the answers, go into the next session. So
I can maybe start just with, because mine is very short, and I was really interested in your work, and what you're doing can actually be applied. We can take that information, and since we are already mapping using the OHT201, we can probably extract all that information and get it into methods to AOP. We just need to have Jeannie or the ones on the other side incorporate into to the knowledge base. But so
Yeah.
we have used some examples for when we developed this model, but we haven't just to get the information out, but we haven't used a specific model for it in that sense. But we have looked at different AOPs in the wiki and looked for those methods that were in there. So we had a case study where each of us that were doing it were taking our favorite AOP, and then we were trying to map it and try to see how the information would come out. And I don't know if Jeannie will talk something about when we used she developed a prototype where we actually tried to incorporate those things into the model as well. So I don't know if you will mention that in your talk later, Jeannie. Then we can.
Unfortunately, I don't have enough time because we have had so many speakers, so that I won't be able, I'm not going to cover a lot of the details, but what I can say is that one of the challenges, and I really like that we got to have the two of you speaking back to back, is that when we were in Methods to AOP speaking very broadly about methods, and I think Madison highlighted this a little bit, like ToxCast assays fit really nicely along with an MIE, but in Methods to AOP, we were often talking about test guidelines is what some people were considering as a method, right? So then that becomes, we need even more granular ways to break down what are the different data objects and what are the properties that we're going to have on each data object. And so it's still kind of a work in progress. Having a single assay, a single table and data model for assays is important, but that is tricky to figure out. So yeah, I don't know if that's a satisfying answer or not for you, Helena.
Yeah, no, thanks. And I see Ellen also adding here with the tox temp that could be added. So we're using the tox temp for all of the DNT in vitro battery assays. And I agree, we have also started to use those to incorporate some of the OHT201 information because it's not always available from ToxCast or ICE. So yes, there's more, and we are also working on getting them using large language models to extract the information. So yes.
So I guess to just a little bit more about the GD211 effort. So yeah, a lot of the fields that are, I guess, requested for the OHT201 could be directly supplied via the curation that we're doing for the GD211 effort. I guess one of the limitations with past GD211 efforts, such as the tox temps, is that, you know, you're just creating these standalone flat files, which is definitely not ideal. So I think one of the advantages of what we're doing is we're databasing all the information. So if folks want to read this, you know, a human readable report, we're generating that, but all of the underlying fields that are used to generate this port are just available within InVitroDB. So you can, you know, grab it that way and do your string searches or whatnot. And then I don't know if there's other questions or if that helps.
Do you want me to read you the questions that were for you, Madison?
Okay, so for the one about, so for the one about the assay AOP mapping, so I'm not the person to ask about that. That would be a Travis and Dan question. It's expert curated. And I guess one of the requirement is that they're only looking at the OECD approved AOPs. I'm not too familiar with the AOP wiki, so I'm kind of leaning on them for doing the mappings, but happy to, I guess, help from the assay perspective where needed. And then I don't know about, I haven't necessarily dug in too deeply into the assays that do not have AOP mappings. That's definitely something we can do because, you know, a lot of the assays do not have coverage, I guess, but they might have, I guess, provisional AOPs or whatnot. That's just not going to be something that we're including in the database at the moment for this next release, but it could be a future effort, I suppose.
Okay, well, do you have more questions there? I don't think so.
I think that's all.
Okay. Okay, well, thank you so much to the speakers. And before we move on to our next speaker, Ann Tesson from UNC Chapel Hill, I just want to put in a plug for a social gathering tomorrow at 5:30 in the RTP box yard, where you can continue your conversation. Thank you, Ann. The podium is yours.
Okay, hello everybody. Let me pull up my presentation here. I'm hoping everyone can see the slides and hear me. Okay. All right, so I'm going to take a 30,000 foot view here and take a step back and start to talk about the data infrastructure that the AOP Wiki and the entire ecosystem of AOP tools and services could fit into. So again, my name is Ann Tesson, and I'm going to speak today about ontology to support the Human Exposome Project. So the Human Exposome Project was launched during the Exposomics Moonshot Forum that happened about a month ago. And it happened with the release of the Washington Declaration. And you can go online and find this thing. Here's a excerpt from the text, and you can see that both ontologies and data infrastructure are specifically called out there. Now, I like to think that I had a lot to do with that because I was involved with the planning for all the Exposomics brainstorming, but all throughout the development of these programs, two themes kept coming up, and that's the issue of data heterogeneity and data linking. And when you're talking about exposomics, especially G by E equals P, you know, the genome and exposome equals the phenome, you're really talking about everything. And that kind of sounds like a boil the ocean kind of problem, but there are a lot of existing knowledge bases that we can use to get a really good start on this sort of thing and do something, put together something really valuable. And the AOP Wiki ecosystem, I think, is one of these. So in my opinion, the Human Exposome Project would be best served by an ontologically supported knowledge graph. So this talk is going to focus on ontologies and knowledge graphs and how they fit together to create a data infrastructure. And so I'll be discussing the existing infrastructure and what updates are needed in order to serve the Human Exposome Project. I'll also briefly touch on how the AOP Wiki is one of the many important data resources available and how it could fit into this infrastructure. So first, let's take a look at the Monarch Knowledge Graph. So the Monarch Knowledge Graph integrates a bunch of different data sources that include information about genes, diseases, phenotypes, anatomy, etc. It does integrate data across species, and you can interact with this graph at monarchinitiative.org. So you see these data sources here in this column. I'm assuming you can see the arrow as I move it on my screen. I hope so. Each of these data sources maps its data onto an ontology or a standard that you can see here in this column. And then these ontologies are mapped to a unifying ontology like Mondo for diseases or UPheno for phenotypes. And that's how all this information gets integrated into a larger knowledge graph. So ontologies and knowledge graphs they're very similar, but they differ in a few important ways. Ontologies tend to be very hierarchical and authoritative, and they move very slowly with their updates and changing things. The kind of information that you would see in an ontology are things like a femur is a part of some leg, right? So these are things that are true of all femurs. The knowledge graphs, they move much more quickly. They are less authoritative, and there are far more tools and services available for working with and creating knowledge graphs. The kind of information that you might see in a knowledge graph would be that this particular femur is 12 centimeters long. But you can also combine ontologies and data about specific data points, like I just said, into a knowledge graph. So a graph is a great way to combine those two different, more generalized and more specific data types. In the Monarch knowledge graph, we take advantage of this in order to do cross-species inference. So for example, you can do gene orthology across the graph using PantherDB, or we take advantage of anatomical homology via Uberon. And my personal favorite is the cross-species phenotype similarity via UPheno. So that means we can look at collections of phenotypes that, say, are associated with different diseases or that are associated with different patients, and use fuzzy matching in order to, say, diagnose a patient with a particular disease. And the Monarch knowledge graph forms the basis of some important diagnostic tools, particularly Exomizer, which uses these sorts of semantic similarity analyses to identify causal variants or genetic variants. But if you look at this, you'll see that the environment and exposures are largely missing. And so that means that any sort of clinical applications that the Monarch knowledge graph is supporting would not be very helpful for someone who has a disease that has a very strong environmental component. And so what I would like to see is a unifying ontology here that would cover exposures and would link data from data sources such as AOP Wiki, and be able to pull those into this graph so that we can do more applications with diseases and phenotypes that have a significant environmental component. So I'm going to look at the ontologies that we have right now for representing exposures and for representing diseases. So first we have the environmental conditions, treatments, and exposures ontology, or ECTO. The structure of ECTO is based on feedback from several toxicology-focused workshops, and the structure is based on the exposure ontology, and you can see the diagram here. So according to the exposure ontology, an exposure event is made up of three parts. You have the stressor or the stimulus, the receptor, which is the thing that is being exposed to the stressor, and then the outcome, which would be like the disease or the phenotype or the endpoint. And each of these elements has its own suite of very important metadata. This ontology has already been used to analyze the PEGS data set and look at interactions between diet and female reproductive disease. And ECTO is also part of the Obo Foundry interoperable suite of ontologies. So that means that for representing stressors, such as chemicals or environmental materials, we would use terms from other ontologies to compose these exposure events. Same for outcomes. We would use ontology terms from the disease ontology or a phenotype ontology. So while these all these different pieces of metadata are very important and they are all included in ECTO, it does make ECTO more complex and thus a bit harder to use. The Mondo disease ontology is a unifying ontology. That means it brings together several different disease ontologies and terminologies. It includes over 25,000 diseases, and it also links diseases to their cause. Links to phenotypes are made in the graph. So I do want to emphasize that there are two different places to make assertions. You can make assertions in the ontology, and you can make assertions in the graph. And one is not always appropriate for the other. Most of the causal links that are in Mondo right now are genetic, but there are a few environmental, about 128. So this is a graph showing the different terms in ECTO that are used in Mondo in that way. But one thing that we noticed when we were looking at Mondo and ECTO and how ECTO is used in Mondo is that precise methods for linking diseases and exposures are not consistent. And this is particularly a problem with social and behavioral exposures, mostly because there's no one clear ontology that represents all of these and that would be clear that we should use. for this purpose. There are some exposure-related diseases in Mondo that are not linked to their exposure, and one example is farmer's lung disease. So after looking at the current state of linking between exposures and diseases, we decided to try to add a little bit of exposure data to the Monarch Knowledge Graph to see what we could learn through this exercise. So we chose the Micronutrient Information Center at Linus Pauling Institute at Oregon State University. The MIC is a website that made up of several different web pages, and each web page is about a different, the health impacts of different nutrients, minerals, or other dietary factors. So using we made a LinkML schema, and we used OntoGPT to harvest information from these free text web pages and added over 1,200 relationships to the Monarch Knowledge Graph. Sierra Moxon's talk later today will talk more about LinkML, and Harry Caulfield's talk tomorrow will talk more about OntoGPT. So you'll get more information about that later. Just know that we were able to get over 1,200 relationships linking dietary factors to diseases, phenotypes, other dietary factors, things of that nature. Now, some of these assertions were suitable to encode into Mondo. I think I would probably get some pushback from Mondo curators, and some relationships were definitely suitable to encode as relationships in the graph. So we got things like vitamin A causes hypercalcemia, and vitamin A prevents xerophthalmia, things of that nature. And these were all new relationships. So there was definitely an information gap there. So after doing all this, we sat down to think about what are the gaps. So knowing what we know about the needs of the Human Exposome Project, for example, we need an infrastructure that can integrate exposure data and knowledge across species, discipline, granularity, terminology, etc. With that in mind, what are the gaps that we need to fill in the semantic infrastructure in order to get there? Well, we need a unifying ontology for exposures. And Ecto can do this with some updates. Like I said, right now it's very complex. We can simplify it so that it'll work. It'll be easier to use for this purpose. We need to update the knowledge graph data schema, which is in LinkML, to support exposures. So this would be extending the Monarch knowledge graph. And we need to sit down and have a discussion about what information would be appropriate to encode in the ontology versus in the graph. And then we need to make repeatable pipelines for creating machine-readable relationships from existing data sources like AOP Wiki. And I will say that this is community work. This isn't something I can just go away and do. It just wouldn't get used. So specifically, how does AOP Wiki fit in? I took one look at AOP Wiki and I instantly thought, well, these are GoCams. And no, I did not conspire with Jim Balhoff to make this slide. I just noticed yesterday that Jim was talking about this. So that's great. We're all on the same page. But as I was looking around AOP Wiki, I did notice that it will probably take some work, but it won't be impossible. Of course, automation is the goal. We want to be able to find an automated way to do this. We don't want to have to sit down and manually map everything. And there are different ways that AOP Wiki are different from GoCams that make it so that it's not just going to be a simple one-to-one. One example I found was in AOP Wiki, it says that paraoxon is adjacent to acetylcholinesterase inhibition, whereas the GoCam version would say paraoxon inhibits acetylcholinesterase activity. So the inhibition part of that is in the relationship in the GoCam, and not in the AOP Wiki. So some discussion is definitely needed, and I'm sure Jim Balhoff's talk will pick up where I left off and be much more informative. Like I said, I did not talk to Jim before making this slide, so he might totally contradict me, and that's okay. You should listen to Jim, not me. But the great thing, if AOP Wiki does get transformed into GoCams, GoCams are made using the Obo Foundry family of ontologies. So that would make them sort of instantly compatible with just about anything that with other things that also use the OBO Foundry family of ontologies, like Monarch Knowledge Graph. So in conclusion, I would love to bring information like what's in AOP Wiki into the Monarch Knowledge Graph, into the larger biomedical data infrastructure. And I think we can do this by making ECTO a unifying ontology. And it's more than just AOP Wiki, it's all the data sources that use all the other exposure ontologies and vocabularies. And so we can finally bring the exposome into this data environment. And once we've done that, then all that information becomes part of the graph. And in addition to doing cross-species phenotype similarity, we could also do cross-species exposure similarity, where we have different collections of phenotypes, and we can say, well, what environmental exposure could have been important for this particular suite of phenotypes. And that's what has me particularly excited. And so finally, exposure data will be part of the larger biomedical data ecosystem, which I think is really cool. All right, and with that, I can take questions whenever it would be appropriate.
At the end of the session, yes. Thank you so much, Anne. We have David Hines next from RTI International. David, please.
All right. Hopefully you are seeing my screen and hearing me. All right. Thanks for the opportunity to present to you today. My name is David Hines, and I am a computational toxicologist at RTI International in Raleigh, North Carolina. I'm going to be talking about some work that we've been doing to improve the ability of researchers to relate and understand data using mechanistic frameworks across the source to outcome continuum by leveraging data standards. And I'm going to be presenting the greater context of this work at a high level and highlighting how we're using standards to strengthen AOP-based analyses to support decision-making. But the next talk by Sierra will get into some more of the details about how we're implementing these ideas into BioLink model, which is a LinkML-based framework to support our analyses. And also this work was funded by NIEHS and is done in conjunction with Chris Mungall and Sierra Moxon's group at Lawrence Berkeley. All right, so I want to start off with a high-level view of how data and metadata standards support the data that we collect and why that's so useful. So when we think about standards, we're thinking about ways that we can make the data consistent, reported in a manner that's clear and useful, but also machine readable. So on the data standard side, that's things like semantic standards, like controlled vocabularies and ontologies, but also reporting standards that guide what information is reported, like the minimum information about microarray experiments, and syntactic standards that define the structure and formats of those data. And on the other side, then there's metadata that are needed to properly use the data we collect because they provide the underlying context where those data come from. So that's things like the descriptive metadata that captures methodological details like cell line or the species used, and then structural or administrative metadata also. So there's really two pillars of this bridge that are holding up our data, and both of them are really important. And the reason why that's really important is that we want to make decisions in multidisciplinary fields like environmental health sciences, and we often need to consider multiple information streams. So, for example, you could consider human health outcomes to occur at the intersection of biological, social, and environmental conditions, each of which might be observed using different mechanistic tools that individually are difficult to draw inferences from, but in concert, you can use them to inform human relevant outcomes. So being able to identify, understand, and integrate data sources is really key to improving what we can say using our data. And of course, the problem is that the data we have available are often fragmented and lack those contextual metadata. So in environmental health sciences, for example, we have diverse domains all along the source to outcome continuum that range from exposures all the way to phenotypic outcomes and everything in between. But those data are often siloed and they're missing critical information. So for example, you might gather in vitro data related to an established adverse outcome pathway, but without understanding contextual information like what cell type was used, what were the experimental conditions, how was the dosing administered. It's really difficult to confidently support conclusions about pathways along that source to outcome continuum. So I've used that term source to outcome continuum, and that is a primary component of this project. So I want to take a minute to clarify and describe exactly what I mean. When I say the source to outcome continuum, we're thinking about the totality of movements, interactions, transformations, and biological responses that lead from the introduction of a substance into the environment all the way to observed adverse outcomes at the organism or population level. So for example, a manufactured chemical might be incorporated into a product. It might enter the body by inhalation while using the product, and then reach concentration in the tissues that activates toxicity mechanisms, which we represent through an adverse outcome pathway that could lead to a toxic effect. And of course, layered on top of that is the complexity of demographic factors like age, geographic location, occupation, school site, and all of those are going to impact the exposures as well as phenotypic responses that occur. So we're interested in this project in improving standards such that information collected from different components along this continuum can interact with and inform adjacent components so that researchers have a standard to guide what information they capture, and that information is harmonized and interoperable such that we can say more about potential impacts from the data we collect. So in this project, we'll be using the existing adverse outcome pathway and aggregate exposure pathway mechanistic frameworks, and we'll be linking them together to describe the domains in that source to outcome continuum. So AEPs and AOPs are largely parallel frameworks. The components are laid out along the top of this figure. Aggregate exposure pathways describe fate and transport, external exposure and internal exposure, whereas the adverse outcome pathways kind of begin with that internal target site exposure and a molecular initiating event, and then describe the causal mechanisms that lead to adverse outcomes. We're trying to advance standards and terminologies and build a foundational infrastructure for data interoperability, findability, and reusability. Our approach for doing this is to develop a data model to harmonize experimental data and outputs across the source to outcome continuum, and we'll be implementing changes by expanding the BioLink model, which Sierra will say a little more about in her talk, which is coming up next. But really the goal here is that we need to identify data needs across environmental health sciences, and we need to implement those standards and then test them in a real world use case example, because as some of the previous speakers have pointed out, there are nuances when we go and actually try to implement adverse outcome pathways to inform decision making. And so in this work, we're specifically going to be using a use case of PM 2.5 exposure leading to decreased respiratory function. And we're going to be using that really as our example across the whole source to outcome continuum. So we're linking together models across environmental health science subdomains to implement and test standards. So on the exposure side, we're using an interpolation model of EPA's air quality system for North Carolina in conjunction with RTI's synthetic population model, which is essentially a scrambled census, to create an exposed population that reflects real-world variability. We're then using deposition models to link those external exposures to internal exposures, and then adverse outcome pathway models to lead from internal target site exposures to our clinical outcomes. For the purposes of this talk, I'll be focusing on the AOP side of the source to outcome construct, and specifically how data and metadata standards can support AOP discovery, validation, and application. So here we're looking at AOP 148 from the AOP wiki, which we use as a starting point for our use case. So here you can see there is a molecular initiating event of EGFR activation, and that leads to several key events, including SP1 activation, goblet cell proliferation, increased mucin production, and mucus hypersecretion that spans several levels of biological organization. And so this is a useful test system to evaluate the interoperability of multiple data sources. But also, when you look at this adverse outcome pathway, and think about it in the context of an application, you immediately see that there are some areas where greater clarity is needed in order to operationalize it and draw some conclusions. So for example, what do we mean by decreased lung function? Are we looking at asthma or COPD? And when we start to consider some of these more specific outcomes, it quickly becomes apparent that not just any data that inform on these key events in this adverse outcome pathway are going to be applicable to every situation. So that's why it's really important for us to build and consider functional real-world scenarios so that we can understand better how to capture and communicate that information that's needed. So for example, our experimental exposures, acute or repeated, that's important when we're thinking about asthma and COPD. Were data based on in vitro responses? Were there in vivo exposures? And if so, what species were used? So we need to be able to identify and capture the data and metadata standards that can ensure that this data can be assembled in order to support AOPs that, and when applied, we can select which data are most relevant to use. So standards that help to ensure data adhere to FAIR principles and also provide that context for data are both really necessary and useful because they allow us to leverage emerging technologies like machine learning and AI to assemble information and verify or even potentially predict adverse outcome pathways. And also, interoperable data can support quantitative analyses across multiple studies and can enable different domains to utilize data and knowledge to extend their work and to build knowledge and improve our inferences. So that really brings us back to the bigger picture of how data and metadata standards across the source to outcome continuum are a huge benefit to understanding what our data can really tell us and to getting more out of the adverse outcome pathways that we have by linking them to exposure data that can inform on human relevance. So for example, if we expand the utility of the data that we collect by allowing the assembly of data into linked models that can describe an adverse outcome pathway and then relate them to those sources of contamination across the source to outcome continuum, we could definitely have some better inferences for our decision-making and risk assessment processes. So briefly, because I think I'm low on time, the take-home messages are really that there's a great need for consistent application of data and metadata standards, and that if we're able to do that, it can greatly improve the utility of the adverse outcome pathways that we have, including increasing machine readability, which, as we've heard from other speakers, is really going to bolster our ability to leverage and make use of these AOPs. And finally, that we need to be able to harmonize our standards across the entire source to outcome continuum to really be able to make the most of our AOPs with regard to risk assessment and decision making. So with that, I think I've gone over. So thank you for your time, and I'll hold questions until it's appropriate.
Thank you so much, David. And our next speaker is Sarah Moxon from Berkeley Law. Yes, we are running a bit behind, so please keep to your allocated time. Thank you.
All right, I will do that as much as I'm able here. Let me get my presentation up. Hopefully you can see it and you can hear me. As I was introduced, my name is Sierra Moxon, and I am a staff software developer at the Lawrence Berkeley National Lab. For this talk, I'm going to be focusing on the project I work on with David, who just presented, about making standards between the source to outcome continuum. But I also work on a lot of projects, including the NCATS Data Translator project with Anne, and the gene ontology with Jim, who you've heard from and will hear from soon. And so I think we can draw some parallels from a software and modeling point of view between all of these projects. And I will talk a little bit about how we do that. So as folks have already mentioned, I think throughout this talk, if as software developers, if our data looked like this with clearly labeled attributes and whole numbers and harmonized units and everyone using the same thing, it would be awfully boring, but a lot easier. But in fact, all of our data across many of the different projects I work on looks more like a spaghetti monster, right? So we have domains that are not static, data collection happening in many different parts of a biological system. Our identifiers tend to proliferate, so it's hard to map one source to another. Sometimes we don't have enough documentation from one aggregating database into another, so we don't know what the provenance is of that data and how it carries through, or even sometimes what the columns mean in a data set. And we know that in the middle of all that spaghetti are answers to some of our really tough questions. And this is exactly the problem that we've been facing at the NCATS Biomedical Data Translator. We knew that we wanted to find the answers to questions that span a number of different biological domains. We know that there are, hundreds and hundreds of sources of data out there. If you look at something like fairsharing.org, there's almost 3,000 databases, 1,500 standards, all these different ontologies. And in further to make our lives difficult, we were also a federated team of developers, right? So we had different kinds of code bases and different kinds of graphs that we wanted to bring together. And so very quickly, we realized that what we needed was a communication layer, a model that we could distribute between all of our teams and also have it talk to each other so that we could have an integrated graph, as Anne mentioned, of all of these different data sources brought together. And if you look at the project we're working on with David, this is the same sort of situation in environmental health sciences. Our data spans very diverse domains like toxicology and exposure, like David just mentioned. We sometimes have siloed data sets that are inconsistent, or we don't understand the metadata produced with them because they don't have a model distributed with them. Sometimes there's missing context. We do a lot of things right, but it's also kind of like pointing to the difficulty we have with reusing data. And so instead of the spaghetti monster, what we want to do in this project is see more of a nice ribbon that moves us from exposure all the way into outcome. And we can see, as David already mentioned, all of these different kinds of data sets we can use to test whether or not the model that we have for communicating between all these different quote unquote silos actually works. And, as Anne mentioned, there are a lot of ontologies out there, Ecto being a very good one and one that we should consider heavily for our project as well. But if we're talking about population demographics, we might also want to include some other ontologies available in the Foundry. And if we're talking about the outcome, two sides of this continuum, we might want to look at HP or Mondo as well. And so how, as data producers, do we incorporate all of these ontologies together to make a model that we can use outside of one particular project? And I think what Translator found when they're doing this for a different kind of data, but the same sort of activity, is that making a centralized model helps us understand when we're bringing data into that particular system, how to map it to other data in the system already. And so what we developed from Translator and from our work there is a model called BioLink, and it's based on the LinkML standard. LinkML is a data modeling framework. I think the important things to remember there, I don't have enough time to go into all of the details of LinkML, but contact me offline. I'm happy to talk about it more. It's one of my favorite things in the world. I'm a lead developer there. But what the main things to take away from this is that LinkML provides sort of like a overarching ontology or overarching data structure that you can use outside of any specific data storage technology, meaning I can distribute BioLink model in JSON. I can distribute BioLink model in SQL for a SQL database. I can distribute BioLink model in an ontology format that we can browse at BioPortal or the ontology lookup service, just like an ontology. And I can distribute BioLink model to fit in any kind of technical architecture that you have. It sort of is focused on biologist-friendly concepts, high-level things, genes, diseases, exposures, biological processes. It's hierarchical. It's already being used in a number of different projects. You can read more about it online. We also sort of distribute documentation automatically. So BioLink is made up of classes, but as you can see here from a little description of what BioLink does, it's highly annotated. We tend to value bringing together researchers of different domains, so scientific software developers, subject matter experts, data analysts. We tend to really value bringing those together and documenting their expertise in a central location that is technology agnostic. So here you can see some of the foundations of the language. In BioLink, we can provide detailed descriptions, human readable descriptions, aliases to other kind of domain specific languages. When we represent a particular concept, we can always link to those. We can also map to external ontologies so that we're reusing the knowledge that already exists there in a way that's very relevant and computable to the classes we're making in BioLink. And we can also sort of give a clue to people using our model as to which ontologies to use. So for a particular disease class in BioLink, if you want to make a disease object, you should probably use identifiers from Mondo to represent that. It also has a whole lot of attributes for those classes. They follow the same sort of rich metadata that the class structures use here. We have subject matter experts that add descriptions to these. So a very simple attribute like name could have a description written by an expert or from a software engineer in this case. And they have mappings to other kinds of names in our space. And that can be really helpful, right? Like you can make a very simple subject, predicate, and object seen here on the right. The green balls are classes, and the line between them is an edge. Those green balls are also nodes in a graph, edges in a graph. And you can make very simple statements that allow you to put a schema on your knowledge graph. So I can say that a chemical entity affects a gene, and all of that's represented in the BioLink model. But BioLink also allows you to get a lot more specific and a lot more detailed by putting together all of the elements in the schema in a more of a less nested or layered approach. So in this example, I'm trying to represent the statement, hexachlorobenzene metabolite increases the methylation of the mutant form of a CDKN2A promoter in the nucleus of a HeLa cell. And by combining all the different parts of BioLink together, you can actually express that statement as well. Don't have enough time to go into this in detail, but I'm happy to speak to questions afterwards. So for our particular project with David, we're actually taking what you can see as these individual data sources and starting to map them up to existing classes in BioLink model so that it can fit in with other work that we do that uses the model. You can kind of start seeing that, maybe an exposure can be tagged as a BioLink exposure event, right? And a population from our synthetic population data could map to a BioLink population of individual organisms. The environment could be mapped up to a BioLink geographical location. And then at the very end of the continuum, we could have BioLink phenotypic features and diseases. And if you remember from the previous slide, these are classes and they have relationships between those classes. So we can start to see that an exposure event might have a connection to a particular chemical, right? The chemical that is being exposed to. And we can have properties in BioLink model that describe that relationship. And finally, that also lets us see where you could apply some of these ontological terms to those larger classes and have a mapping between the kinds of data that we see in practice and the higher level model here that describes what to use. So back in the class slide, I showed that, a disease might need to use a Mondo ID. So you could easily say that, yes, asthma is described by this Mondo ID. It is typed with this biolinked disease. And we start to see a model form from that. And all of this is the purview of the BioLink model. So it's giving you sort of a map to show how you go from these siloed events to a more general layer of classes that can be used across many different data sources, and then specifically what kinds of ontologies and what kinds of identifiers will help us do that. And so I think what success looks like for this particular phase of the project is just being able to say in more detail what each of those areas of the source to outcome continuum are, what kind of classes and slots are used in the BioLink model to describe them, how they're related together. We can also see from this experiment where we're going to apply these standards to synthetic data, where any gaps in our standards exist, we can help contribute back to the ecto-ontology that Anne mentioned, and we can start to incorporate some of these diverse domains into a single place where you could actually find those like insights that we're looking for in all of our different projects. And of course, our next steps here are really to engage our communities, right? We need those subject matter expertise to contribute to the model. We need those software engineers that are trying to do this work of pulling these data together to contribute to the model. And so we have a lot of work to do. And that is it. Thank you to everybody on the call already that was mentioned, and also to the NCATS Data Translator and NIEHS for funding a lot of this work, and the Monarch Initiative for providing such good, valuable feedback into the model as well.
Thank you so much, Sarah. I think that's a good opportunity to ask questions our presenters in the last session. And David and Sierra. So please raise your hand if you have any. And I said there was a discussion, an interesting discussion in the chat, but I don't think we can address it right now. It's too complicated. Ginny, please go ahead.
Well, I don't have a question. I just want to say thank you to the last three speakers, and you've taken some of my punchlines, which is good because we're behind. So just I want to say thank you for those talks. I'll go ahead and get started.
Yours.
Okay.
You should.
Thank you, Maria. Okay, great. All right. So hi, again, I'm Ginny Hench for those who missed the earlier presentation, and we're way behind schedule. So I'm going to be skipping a few slides so I can get you all into breakout rooms so you can have an opportunity to discuss what you heard. The same disclaimer statement I made earlier. So within the AOP wiki data model, there are areas that I would consider having potential for semantic interoperability and areas that currently have semantic interoperability. I already talked about evidence fields as that one is an important field we need to restructure. Other fields that Helena referred to today from Methodist AOP concern how is it measured or detected and the regulatory endpoint relevance. One particular piece of the AOP framework is the event component concept, and this one already offers semantic interoperability, and I'm going to briefly go through how this EC concept was used for text annotation purposes under the EPA's endocrine disruptor screening program, and this will lead into some of the data model stuff that I've talked about. There's a rationale for this diversion. So this slide shows some of the key components of event components without listing out which ontologies are relevant. And I'm also showing phenotype here as with the suggestion that we consider including phenotype as separate from a biological process when we're talking about key events. And this was something that we found to be valuable and not that hard to do when we were doing the manual annotation. Some of the reasons, oh, I removed the slides. Some of what Anne touched on in her talk with relating to some advances that are happening in the phenotype ontology spaces provide the rationale for why I think there's value in covering phenotype. Fortunately, I hid the slide already and I'm not going to try to retrieve it. So what you see here are different entities that we were labeling when doing manual annotation. So we found that in addition to labeling event components, which would cover object, process, and action, the most minimal parts of describing a key event, that also looking at the causal agent, the method of measurement, and the context were very helpful. So in the AOP framework, those other entities are not technically event components, but they provide value. And so if the recommendation that I'm making is that if people are looking at AI approaches or NLP approaches to try to extract AOP information, to consider, all of the AOP entity types. This is an example sentence that we annotated. And the value of this, first of all, it's not very scalable for humans to be manually doing this work. But as a proof of concept, this exercise helped inform the way that we think about the EMOD work and how we can model evidence and objects by using these entities that then are sortable and filterable. And as an example of exactly what I mean, here is an EC table holding text extracted ECs that map to a key event that's already in an AOP in the AOP wiki. And by collecting information in a more automated way, we can keep track of the source article that an observation or line of evidence came from, which can help with provenance tracking, which based on a lot of the work that some of us in the AOP community have done in terms of thinking about how Penny talked about trustworthiness, provenance tracking is important to say, hey, there are people who have done this work and they've used scientific method to demonstrate this is a reliable source of information. All right, so now we are at the EMOD concepts. So to touch on some of the points that Helena brought up earlier. So on the right side, this purple box is the assay data object that we have in the prototype right now, the EMOD prototype. And you can see some of the properties that are listed there. We included detection technology. And I know that Jennifer Fostel has done a lot of work looking at the OBI terms for detection technology, which she will cover tomorrow. And I think she was one of the people who influenced us to include detection technology here. And the other thing I want to highlight is that when you look at the observation data object and the assay data object, both of those have a biological object and a biological process. One thing that is important that I want to highlight is that in the assay, we talk about the targeted biological object and targeted process to emphasize that that there's this potential for a measurement to be collected, but the actual measurement that then comes with some kind of experimental effect is going to be associated with the observation. All right, so another principle I want to explain here with this very colorful visual is this idea of AOP observation evidence roll-up. Yeah, so you see the AOP at the top and the colored bars are meant to represent evidence, stressor, assay, other properties that can be associated with the observation and evidence data objects that are, that are lower in the sort of AOP hierarchy. And the reason this particular principle has been driving the EMOD work, even though we have we're on EMOD version 2.0, we've not yet gotten to this point where the observation and evidence information are available on an AOP page, but that is the idea, that we would improve the user experience by making then people wouldn't have to click through so many pages, but also thinking through and drawing upon some of the themes we heard in from the last presenters. The context of information is really important. And when AOP authors are making an overall assessment of an AOP, they're making judgment calls and talking about pieces of information across all the different elements. And the more we can make that information easier to get to, I think it's going to help with multiple aspects of AOP development. All right, another important concept I want to highlight, or in thinking about that AOPs need to be, serving a regulatory purpose. There are other regulatory entities that we need to think about being interoperable with, and I've highlighted OECD harmonized templates and test guidelines in this schematic. This is not meant to, I'm not trying to suggest that harmonized templates or test guidelines would be stored in the wiki, but more that as data modelers and people who are invested in the AOP wiki data model, that we should think about what's going to make the most sense, what should we store where, and how do we want to make the different related pieces of information interoperable with one another. So the purpose of this slide is to demonstrate some concepts that I think are, this may not all be immediately apparent, but there are some properties that I've included on the evidence observation and assay data object here that were not shown in the previous slide. The point is that we can add what we want to these core data objects, but what's important is to think about what properties are essential and what property arrangements are needed to hold all lines of evidence for AOPs. This particular model, or the last EMOD version 2.0, was heavily dependent upon the way data was modeled for KER 2937, which is a KER for fish. And that, I don't know how well this particular arrangement of properties is going to hold up. And so that is one of the things that I invite all of you to consider in the breakout discussion, and share your ideas and feedback. All right. I'm not going to read through my acknowledgements here, but I do want to acknowledge one person, Crystal Lee Powell-Jackson, who was involved in helping to submit the AOP Wiki 3.0 grant proposal that some of you heard about a few years ago that we were pursuing. Unfortunately, it did not get funded, but Crystal was a great contributor, and I think she has some really great ideas about how AOPs can serve public health needs